and it's called dry not because of our humor, but because we only work with data and no actual extracts and solutions and so on. And there are actually two dry labs at BTI, Zhang Chongfei's lab and my lab. And there are at least a couple of semi-dry labs, I would say, like a good Finger Lakes wine. And so what we are doing in our lab is, we're using genomics and breeding to leverage, the yield of and the efficiency of the breeding process. And I'll go a little bit into that, but I won't bore you with too much details. So the problem is, crop breeding is actually pretty slow. And so with new genome technologies, we can speed up that process and get much more gain. And of course, you all know the human population is growing, everybody's hungry, we need more food in the world, right? And so what we are trying to do is leveraging genomics to accelerate the development of these new lines. And so my lab has been working on this project that's been called BreedBase at some point, and it's a system that allows breeders to manage all their data, to do all their predictions and all their selections. So it's very applied, and I think Mr. Boyce Thompson would like this project. And so one important part, of course, in this story is the genome information that we have for many, many plants now. And for example, the tomato genome was heavily done here at BTI with Zhang Chongfei and Jim Giovannoni and at other Cornell labs. And of course, we have produced a lot of other genomes here at BTI. There are some other important genomes here that were not done here, but just to illustrate that, we have all the genome information. And what we can do is we can really characterize a line that we get from a breeding process, and we can compare it to a reference, and we can tell immediately all the differences, and then we can take these differences and correlate them with phenotypes. So the other important point is phenotypes. So what breeders are doing, they are planting a lot of fields with a lot of varieties, and then they're looking at the plant and they're phenotyping them. An important phenotype is fruit size, for example, so they measure fruit size, but they measure maybe 100 other traits routinely. So it's all a lot of data. So there's a lot of genotypic data, there's a lot of phenotypic data. Now what you can do is you can correlate the genotypic data and the phenotypic data, and if somebody gives me genotypic data from a plant, I can actually predict what the plant looks like. So I don't need to wait and grow that plant. I can already select it and say, that's a good plant. I will, it will go on in the breeding process. And that speeds up the breeding cycle. And so, breeding is like Taylor Swift, we're breeding eras. They're not quite as separate as with Taylor Swift's eras, but in the old era, maybe pre-1990, it's what I call the paper era. And mostly the technology that was used there was phenotypic selection, so people just looked at the plant. And that was the phenotyping that they did, and that's important. That's still being done. But this is usually, if there's no genome data in it, that's usually called breeding 1.0 now. So Ed Buckler here at Cornell introduced this terminology, breeding 1.0, 2.0, and 3.0, and 4.0. And then after 1990, we had the Excel era, so people started to use computers for data management. But Excel is actually, very nice, but when you want to combine data from several years or several locations, it gets very hairy. And where is that Excel sheet from that trial? It's on the laptop that fell down the stairs or something. So that's not good. And so now we are in the database era, and this is the really, you need these databases if you want to do genomic selection, because the data is so gigantic that you cannot keep it in an Excel sheet. So those are the evolution of the eras. And there's of course the 4.0, which is designer genomes. So as we have heard before, CRISPR, we'll be breeding differently using CRISPR and introducing specific mutations and so on. But for breeding 3.0, we still have the so-called breeding cycle. So I won't go too much into the details, but what the breeding cycle means is, a round of selection of plants. And so here we have our breed-based database. The breeder works with the database. He decides, or she, what plants they want to grow, and tells breed-based to create a field layout. And so breed-based will do a randomization, and there are a dozen different randomization techniques in there. So the field can be planted like that, and you can put barcode labels on it, and then you can phenotype it, you can collect data, you can collect samples to genotype. We use these, we work a lot with these pheno apps to collect data in the field, and so that's a system of electronic data capture. And in the end, you can make analysis with all the data that you captured, the phenotypic and genotypic data. You can, for example, use genomic selection to predict the next cycle, right? And so just a number of crops that we're working on. It started all with tomato and the tomato sequencing project, and other Solanaceae that we put in this database called solgenomics.net. And then this breeding really came about by a Gates project called Next Gen Cassava. And cassava is a really important crop in Africa. About, between 0.5 and 1 billion people depend on cassava for calories, so it's right up there with, the important grains. And then we expanded to other root and tuber crops and banana, also with the Gates Foundation. And in the meantime, other people have started using the system, and so we, pretty much any crop has a so-called, what do you call an instance. And I just want to show you one screenshot, and then I think that's my last slide. This is a screenshot from Cassava Base, so the Cassava database, and you see all the breeding locations of cassava in the database. And you can see that this is really a worldwide effort and has a worldwide impact. And this morning when I came in, I saw this little stress ball, which kind of suggests, says BTI on the globe, and kind of suggests a global impact of BTI. And you can also use it to reduce your stress. But I think it's, I thought, well, this is exactly my slide. This is, they're $7, by the way. I just took it and said I will bring it back, but I may not. But I recommend you buy 2, of course, for both heads. And so, yeah, so, the software is widely used now, and we are continuously working on it. It's a very intensive task. There's breeding right now is an extremely dynamic field where there are lots of new technologies coming up, lots of high throughput phenotyping, new genotyping technologies. Everything has to be constantly adapted to, to these new technologies. And so we're looking into how we can help most people, use these new and amazing tools. And I just want to point out that, most of the work, of course, is done by my team members, Chris, Thomas, Amber, Nama, Christine, Surya, Srikant, Tidima, and Isaac, who telecommutes from the Netherlands. And sorry, I forgot to put him here. So, but that's it. Thank you. Yes?
I'm curious about the digitization of phenotypic data as opposed to genomic. How robust is that? How good is that? Can you use that in the model?
Yes. Yeah, I mean, it's used routinely. Yeah, there's a certain system that everybody uses now. These you have to define the traits really well. So we create things that are called trait ontologies. And they explain exactly how you have to measure it, what the scale is that you have to give it in, because obviously if somebody measures in meters and well in centimeters, there will be just a giant mess. So it has all be really well defined. And I think that's one of the challenges of these projects is that everybody has to agree on these things, right? So you have seen the map of cassava breeding programs, and they all use the same traits, and that's totally remarkable. Yeah, so you're putting your finger on a very good point. Quality data is very hard. Like flavor, mouthfeel, those things are very, very hard because there are no good systems to measure them. You can measure certain chemical compounds, and you can say, well, those are all flavor compounds, but when you eat it, it's still disgusting, right? So it's very difficult. But other traits are much simpler, like size and disease resistance is a very clear trait, for example. Yeah. Yeah.
I know that you have dry lab, but if you want to sequence a plant genome, about how long does that take? I mean, is it months?
Yeah, I mean the thing is the sequencing is actually very fast, right? It's a few weeks. It depends how you have to, of course, grow the plant, you have to extract stuff, and then the sequencing process is not that long. But then you have to assemble it, and then you have to annotate it, and that still takes at least a few weeks. And you have to have all the right data for it, right? You have to have all the it's very convenient if you have a lot of RNA expression data to make the gene annotations. So, but in principle, you could do it let's say in weeks. What would you say, Jean?
Maybe I can add.
Yeah.
It depends on what class species.
But I mean, less than a year.
Less than, yeah.
I mean, it depends on what class species. Class that was, they are genome quite different. Polyploid.
Yes, exactly. Good point. Yeah.
Genome size and maize and Arabidopsis are totally different.
Yeah, exactly.
But you also don't have to, once you do one, you don't have to sequence the entire sort of sample and get a snapshot, and that takes just days. So then you can apply that information.
But it's not years anymore.
It's not years. No. It's literally measured, I would say, in months. If it's not a really hairy genome like octoploid or like strawberries octoploid, that throws in a little wrench in your